Warning: the invisible proteins that are erasing human minds. She laughed so hard she slid to the kitchen floor, tears streaking her cheeks, breath hitching like she'd heard the world's funniest joke. 10 minutes earlier, she couldn't remember her street. 2 weeks later, her hands trembled pouring water. By week 6, she couldn't swallow. The MRI looked like someone pressed a colander into her brain. Holes everywhere. Tests for viruses, bacteria, toxins, nothing. Then the team said a word you don't expect outside horror films: Prion. Not alive, no DNA, no cure, yet it spreads by shape alone. Anyone, any age. Rewind. How can a protein act like a plague? Roadmap and promise: what you'll learn. This isn't science fiction. Invisible proteins can erase minds, and we're going to show you how we know. You'll see what prions are through people who met them first. Families who watched laughter turn into staggering then silence. Farmers who burn entire herds because one cow ate what a cow never should. Surgeons who trusted sterilizers that fire couldn't cleanse. We'll trace how a simple fold goes wrong and convinces others to copy the mistake. A rumor carried by shape. Here's where we're going. First, to human stories. The Highlands ritual that fed a disease, then stopped it. Britain's industrial feedback loop that turned herbivores into accidental cannibals. The betrayal that arrives with no warning when a single protein just misfolds. And the awful truth that medicine, trying to heal, sometimes carried the seed. Then the biology. The cellular prion protein we all make. The warped version that templates its neighbors. And why a Nobel-winning idea that a protein alone can infect took decades to prove. We'll show how prions cross species, why some brains resist, and why deer in North America now carry a slow-burning fuse called chronic wasting disease. We'll face the blood question, the cleaning problem, and the detection race. How tests like RT-QuIC push sensitivity into the clinic, with nasal brushing reports around 97% for sporadic CJD in some centers. We'll look at therapies that almost worked, and why almost isn't enough. Finally, we'll talk about what you can do. Practical shields for hunters, patients, and labs. So fear turns into vigilance, not paralysis. To understand the terror, meet the first families who saw it. The Laughing Death, Kuru in the Highlands. The Highlands air is thin and clean, but the grief was thick enough to taste. In the villages of the Fore people of Papua New Guinea, women and children began to tremble. At first, it looked like nerves, hands shaking while husking taro, a wavering gait on muddy paths. Then came the laughter, wrong and unstoppable, like a tick that seized the whole body. They called it the Laughing Death. Within months, mothers couldn't stand, couldn't swallow, and then couldn't speak. Entire lines of sisters vanished. The village rhythm, birth, marriage, mourning, buckled. To understand why, you have to sit quietly at the edge of a funeral they once held. In the late 195s, as an act of love and respect, 4 women and children carried out mortuary cannibalism, endocannibalism. It wasn't grotesque, it was devotion. They prepared their dead so the person would live within the kin. Women and children, by custom, did most of the preparation and consumed the brain and spinal cord during funerary rites. Men often ate different tissues or abstained. Nobody knew the brain could carry a seed that survived cooking and time. Nobody knew a protein's shape could outlast fire. Here's where a small moment makes the big picture concrete. A little girl, no older than 6, walked behind her mother, who had started to wobble. The child copied the stagger, giggling, thinking it was a game. 2 years later, her own walk began to drift. In another house, a farewell feast was held with a new rule: no laughter. It didn't last. Sometimes the laughter took them mid-bite. Outsiders arrived, hauling notebooks and questions. One of them, D. Carlton Gajdushek, stayed. He didn't yet have a villain, just a pattern. Mostly women and children, clustered families, funerals linking to onset, and a delay that made causation slippery. To understand why it seemed to spread but also seemed to wait, you have to notice the calendars. People who took part in the rites didn't fall ill right away. Years passed, 5, 10, 20, sometimes longer, over 30 in rare cases. The incubation made the map look like rumor, not infection. Meanwhile, something else was happening, far away in a dark animal room. In 1966, after years of skepticism, transmission was shown in chimpanzees. Brain tissue from patients with Kuru introduced into chimps eventually caused disease. Decades-long latency confirmed what the villagers already knew in their bones. Whatever this was, it could pass between bodies without the classic signatures of life. No DNA, no RNA, no bacteria to culture, just a corrupted pattern that could. Teach other proteins to fold the wrong way and keep teaching, like an origami template with a bad crease passed hand to hand. Stopping it turned out to be both simple and impossibly hard. Elders, urged by health workers and missionaries, began to restrict the rituals in the late 195s. Here's the ethical tightrope. A practice that bound families to their dead was also carrying the seed that unbound those families. Respect clashed with survival. The community chose life. As the rites faded, so did Kuru. Through the 196s and the 197s, incidents fell, but the disease didn't vanish overnight. Those long incubations still bloomed. Daughters who had last taken part as children began to sicken as adults. It was a ghost that honored old calendars. For the scientists, Kuru cracked a door. A disease that could be caught without a microbe challenged the rules. Guy Dussec's fieldwork laid the epidemiological groundwork. Later, Stanley Prusiner argued the unthinkable. A protein alone could be the infectious agent. The names would come. PRP for the normal cellular protein and its distorted shadow. But in the Highlands, no one cared about letters. They wanted to know who would be left to sing. There's a quiet scene worth holding on to. A family decided to keep their farewell, but changed the last step. Instead of preparing the brain, they buried it under a pandanus tree and sat with the ground. Months later, a visiting nurse returned with a notebook and found the tree ringed with stones, a small promise that grief could take another shape. The stakes were not abstract. Entire villages risked losing their mothers and sisters. The cultural fabric that raised children and held memory together was fraying. The consequence of each ritual, performed with love, was unintended erasure. Viewers should care because the math here isn't sterile. The people who fed the disease were the ones who nurtured the living, and the cost was paid in flavors. Soups not cooked, stories not told, hands not guiding younger hands. What nobody knew at the time was that Kuru's lesson would scale. If a ritual could feed a disease by moving a shape from one brain to another, then any loop, industrial, medical, or accidental, that recycled nervous tissue could do the same. The moment everything changed for the wider world would come when an herbivore ate the wrong meal. If a ritual fed a disease, what fed the global outbreak? The cow that ate the cow, mad cow's industrial loop. While grief settled over the Highlands, a quieter loop began in British feed mills. Cattle, built for grass, were given a protein boost, meat and bone meal rendered from abattoir scraps, including nervous tissue. It looked efficient, almost tidy, but efficiency hit a shape. Somewhere in that chain, a misfolded prion from scrapie-infected sheep or an undetected bovine case slipped into the meal. Each feeding became a lesson in the wrong fold. On a gray morning in Devon, a farmer watched a favorite cow press her head into a fence post and grind. She startled at sounds that weren't there, staggered, then stood rigid, eyes wide. Vets checked for infections and toxins. The lab called back with a phrase farmers had never heard: bovine spongiform encephalopathy, BSE, spongy brain disease. The rendering loop had turned herbivores into accidental cannibals, and the shape was multiplying. The blind spot lived in process tweaks. Rendering had shifted. Lower temperatures, faster throughputs, enough to spare the prions fold. What nobody planned for was how well prions cling to nervous tissue and shrug off heat and chemicals that usually police food chains. Meat and bone meal moved between farms, across counties, into calves that wouldn't show signs for years. Amplification hid inside growth. Then the curve bent up. Confirmed BSE cases in the United Kingdom climbed until 1992, the peak. Numbers turned into bonfires and empty fields. Entire herds were culled. Carcasses burned in lines that smoked into the wind. Export bans closed markets overnight. A farmer signed the paperwork with a shaking hand, then walked to the yard where his kids used to climb gates. He said nothing. He just watched the flames. Meanwhile, something else was happening in neurology wards. Young people, Teenagers and those in their 20s arrived with depression, anxiety, odd sensory changes. They forgot names, got lost on buses, saw shadows at the edges of rooms. Psych consults tried to help. Then gait problems began. Grimacing, jerks. MRIs and pathology drew the link. Variant, Creutzfeldt-Jakob disease, VCJD, tied to dietary exposure during the late 198s and early 199s. Here's a micro story that carries the cost. A 19-year-old sent home twice with stress, wrote in a diary that her brain hummed too loud. By winter, her handwriting shrank to a child's. By spring, she couldn't feed herself. Her parents kept a calendar on the fridge, ticking days since she laughed last. At the end, the calendar was blank. To understand why some exposures leapt species and others stalled, you need the species barrier and strains. The normal prion protein in cattle and in humans shares a sequence, but not perfectly. Those differences create friction that slows templating. Yet prions aren't single shapes. They're families of conformations, strains, that can adapt with passage. Feed a strain into a species with a partial mismatch and, with time and enough chances, a subpopulation finds a fit. Tissue mattered too. Highest risk sits in central nervous system tissues, brain, spinal cord, dorsal root ganglia, where PRP levels are high and seeds are dense. Muscle meat is lower risk, but industrial processing can smear tiny amounts of spinal tissue into mechanically recovered meat. One detail-shaped policy. Risk isn't evenly distributed across a carcass. Policy scrambled, sometimes live on television. In the late 198s, the UK banned feeding ruminant protein to ruminants, closing the loop that taught cows to eat cows. Authorities designated specified risk materials, brain, spinal cord, tonsils, parts of the intestine, for removal from the food chain. Surveillance expanded, slaughterhouses changed line practices. These moves worked, but time lagged them. Long incubations meant cases kept arriving after bans. Trust took longer to rebuild than herds. Across town from a press conference, a butcher pulled all oxtail from his counter. Not because it was illegal that week, but because mothers asked hard questions with toddlers on their hips. He put up a handwritten sign, "We don't take chances." He lost money for months and kept the sign up anyway. Scientists tracked the numbers like seismologists after a quake. BSE peaked in 1992. The curve then fell with a long tail. Human VCJD totals would never match animal counts, but each case felt like a spotlight on a system failure. Leukodepletion was later added broadly to blood processing, and rare transfusion-linked VCJD clusters years after the food peak reminded everyone that once a prion establishes in a host, it doesn't respect calendars. This was the policy lesson the world tried to memorize. Close amplification loops, remove highest-risk tissues, and assume long incubations will test both patients and budgets. Countries reinforced feed bans, trained inspectors to spot line breaks, and funded surveillance that rarely made headlines. Get this, none of those fixes required solving every atom of the misfold. They required respecting the path the shape takes. Don't feed brains to herbivores, don't let high-risk tissues near plates. Watch for quiet drifts in industrial steps that erase safety margins. When you do that, epidemic curves bend. But not all prions come from dinner. Some rise without warning, from inside a single cell that makes a single mistake and teaches the neighborhood. If hospitals can't cure it, the next question is sharp and unfair. Can they spread it? The betrayal within, sporadic and genetic prions. While regulators tightened feed rules, a different kind of case walked into clinics. No travel to Britain, no wild game stew, no surgery to trace. A teacher in his late 60s misplaced chalk, then the way to the staff room. Weeks later, his hands jerked closing buttons. He laughed at the wrong times, then cried without knowing why. Scans showed spongiform change. EEGs grew chaotic. This was sporadic Creutzfeldt-Jakob disease. One protein in one brain just misfolds, and that error teaches its neighbors. The stakes land in that word just. There isn't a violation to confess. Families sit at kitchen tables circling the same sentence. Did we miss something? Often the answer is no. In most prion diseases, there's no identifiable external cause. A single PRP molecule falls into a bad shape, the way a paperclip clicks into a new angle and stays there. And that shape becomes a template, seeded nucleation, recruiting normal PRP, stacking into fibrils that fragment into more seeds. Cleanup systems can't catch up with geometry that multiplies. Here's a micro story that hurts because it's so small. A retired mechanic started leaving notes. Keys in bowl, dog fed, turn off stove, and then found the notes in the fridge. One afternoon, his daughter watched him trace his name on a check, then stop and stare at the pen like it had betrayed him. By the time the diagnosis arrived, timelines were measured in months. No cure, only time, and not much of it. Meanwhile, some families carry a different clock. They know a name: Fatal Familial Insomnia. A woman in her 40s kissed her kids goodnight and lay awake. The next night and the next. Sleep splintered into glassy pains, brief flashes that didn't restore anything. She started sweating through shirts, her pulse racing while she sat still. Words slipped. Doorknobs fumbled. Her husband counted the hours she didn't sleep and then stopped because the number didn't help. In genetic prion disease, the betrayal isn't exposure, it's inheritance. Mutations in the PRNP gene change. Posit landscape. 1D178N is infamous. It swaps an aspartic acid for asparagine at position 178. On its own, it doesn't write destiny. Pair it with the codon 129 background, methionine or valine, and the course shifts. D178N with methionine at 129 usually leads to fatal familial insomnia, where the thalamus, the brain's sleep gatekeeper, atrophies and nights unravel. D178N with valine at 129 tends to steer toward familial CJD, a faster dementia that echoes sporadic disease. Same mutation, different backdrop, different nightmare. Gerstmann-Streusler-Scheinker disease draws another path. Certain PRNP mutations seed an ataxic syndrome first. Balance gone, steps wide, speech slurred, then dementia. Pathology finds amyloid plaques built from PRP. It moves slower than sporadic CJD, but it moves. Families learn to measure hallways and handholds and to keep chairs near doorways. Genetics sharpens and complicates choices. A brother sits in a clinic with a consent form for testing. A positive result won't bring a treatment, only a shadow over birthdays and job offers and decisions about children. A counselor says, "You're more than a gene," and means it. He nods, then asks the practical question that bridges science and steel. What happens if I ever need surgery? History answered painfully in earlier eras. And hospitals now carry Prion protocols, so instruments don't become messengers. Molecularly, sporadic and genetic cases meet at the same crossroads, templated misfolding. In health, PRP sits on neuron surfaces, mostly alpha helical, probably helping with signaling or protection. In disease, it shifts toward beta sheet, builds amyloid fibrils, and those fibrils break into seeds that spread the fold. Energy landscapes rule, how easily a protein falls into a valley, and how deep that valley is. Mutations can lower the barrier to the bad valley. Seeds can nudge neighbors over the edge. Timing is ruthless. When seeds first appear, symptoms haven't. By the time a clinic notes jerks or insomnia, the city inside the brain has already rerouted around sinkholes. Diagnostic tools caught up faster than treatment. Assays like RT-QuIC can detect seeding activity in cerebrospinal fluid, and in some centers from a simple nasal brushing for sporadic CJD, with reports around 97% sensitivity. Speed brought clarity, vital for care and for instrument handling, but clarity arrived before a drug. Here's another microstory. A mother with a known family mutation writes letters to her kids labeled for future years. Open at graduation, open when you can't sleep, open when you're angry at me. She labels one open when you're ready to forgive me for not telling you sooner, then tears it in half and writes another labeled open when you need to be brave. She never finishes the stack. Emotionally, the terrain is brutal. In sporadic disease, loved ones search for a mistake that isn't there. In genetic disease, a surname becomes a diagnosis you can carry for years before anything hurts. People decide whether to have children, whether to change careers, whether to tell siblings. The health system offers counseling and monitoring. It can't offer a cure. Yet there's agency and precision. Early palliative care shapes days. Speech therapy before words scatter. sleep support before night's fracture, pain control before panic. Families who know their risk can bank decisions ahead of time. Advanced directives, research registries, conversations that trade secrecy for support. Hospitals can pivot faster, single-use tools for high-risk procedures, Prion-specific decontamination when suspicion is high, RT-QuIC fast tracks that spare steel downstream. The betrayal within isn't fate's poetry, it's physics. A fold finds a valley and teaches it. But choices, clinical, personal, and procedural, still draw the boundaries of where that fold can travel. And that brings us to the rooms where care itself once carried seeds. When medicine hurts, iatrogenic transmission. Hospitals save lives for a living, which is why this chapter lands like a punch. Care once carried seeds. Start with a promise that felt like a miracle in the 196s and 197s, human growth hormone for children who weren't growing. It came from cadavers. Teams collected pituitary glands, pooled them, purified the hormone, and injected hope into small arms. Years later, letters started to land like stones. Young adults who'd received treatment as kids began to stumble, jerk, and slip. The link was brutal and specific. Creutzfeldt-Jakob disease acquired not by food or fate, but by care. Worldwide, roughly 226 cases traced to cadaver-derived growth hormone, with attack rates tied to how rigorously each country's batches had been purified. Parents held boxes of childhood syringe caps and asked if saving inches had cost a lifetime. Meanwhile, in operating rooms, surgeons patched skull defects with dura-mater grafts, tough membrane harvested from human cadavers. It was the right material, the right shape, and for a while it seemed safe. Then clusters appeared years later. About 228 cases worldwide linked to those grafts, many tied to a popular brand processed in ways that didn't fully inactivate prions. A woman who'd finally fixed daily headaches after a repair started veering on sidewalks. Her husband noticed one shoe wearing down on the outer edge. By the time anyone connected lot numbers, the incubation clocks had already run long and quiet. There were smaller, haunting parables. An EEG technician in the 197s reusing depth electrodes. A corneal transplant from a donor who didn't have a diagnosis yet. A handful of suspected transmissions from neurosurgical instruments that had once touched high-risk tissue. In each case, certainty bent under time. Incubation stretching a decade or more. Medicine changed protocols, but calendars kept writing outcomes already set. Why here? Because prions are stubborn in ways surgical routines weren't built to fight. They cling to steel like memory. Standard autoclaving, reliable steam and pressure that drop bacteria and scorch viruses, doesn't guarantee prion inactivation. Temperatures that melt plastic won't reliably erase the fold. Some disinfectants cross-link proteins, fixing seeds to surfaces. The fix wasn't better scrubbing, it was different thinking. Picture a sterile processing unit after a near miss. A pathologist calls upstairs, suspected prion case. The OR manager stares at a tray log and a bin of instruments scheduled for tomorrow. She lifts the whole set and drops it into a red-labeled container marked for destruction. Thousands of dollars gone in a clatter. She signs the loss and sleeps that night. Guidelines evolved with scars. When prion risk is on the table, certain neurosurgeries, posterior eye procedures, any work likely to contact high-titer tissue, hospitals isolate instruments, use single-use where possible, and, if reprocessing is unavoidable, follow prion-specific protocols. Long soaks in strong sodium hypochlorite or 1 to 2N sodium hydroxide, followed by prolonged autoclaving at higher temperatures. Even then, many centers choose destruction over reuse. Color-coded tags, prion-labeled bins, and laminated algorithms live at eye level, so a well-meaning tech doesn't drop a suspect wrongdoer into a standard washer and contaminate the chamber. Timing matters too. Dried organic residue shelters seeds. Teams chant a mantra. Keep it wet, get it down, flush lumens now. Not all news stayed grim. The switch from cadaver-derived growth hormone to recombinant synthetic hormone ended that entire iatrogenic stream. Synthetic dura replaced human-sourced grafts. Vendors began flagging prion inactivation compatibility for devices. Education moved upstream. Residents learned prion pathways the way they learn hand hygiene. RT-QuIC brought speed to diagnosis. letting infection control trigger instrument containment early instead of after a biopsy return days later. But lagging clocks still deliver letters. A patient in his 30s opens mail from a National Health Service 15 years after a dural repair. We are writing to inform you of a potential exposure. He reads it 3 times, then sets it face down. There isn't a test to certify safety, only surveillance and a card to hand, future surgeons, so they can choose tools and protocols that don't turn one mistake into many. Here's a micro story that shows what learning looks like. A Tuesday at 2 a.m., ophthalmology flags a posterior eye case with an atypical history. The charge nurse slaps a red Prion precaution tag on the cart. The scrub tech swaps in single-use blades. The reprocessor tapes off a shelf. The supervisor schedules a 134 degrees Celsius extended autoclave run for any reusable that crossed the field. The next morning, the department asks about cost. The supervisor slides a 1-page incident sheet across the table, cheaper than a letter to 200 patients. Impact ripples, ORs pause rooms, schedules reshuffle, finance signs off on losses measured in trays, not tools. Most notified patients will never get sick. True exposures often vanish under scrutiny, but anxiety ignores nuance. Hotlines open, nurses repeat careful sentences. What we think happened, what we did, what you should tell providers in the future. The lesson is precise. Prions exploited our best intentions by writing pooled biologics, graphs, and steel. We answered with different materials, rewritten playbooks, and the discipline to throw away what we can't trust. Perfect protocols only protect tomorrow, not yesterday. But tomorrow is still everything for the next patient. What a prion really is. If care once carried seeds, then we have to ask what the seed is. Not a microbe with genes, not a virus with a script. The infectious agent here is shape. Start with the protein we all make, the cellular prion protein, PRP. In healthy neurons, PRP sits on the outer membrane, tethered by a lipid tail, folded mostly into alpha helices. It likely helps with signaling or shield cells under stress. Nothing sinister. But under the wrong conditions, some PRP flips into a beta sheet, rich form that stacks into long amyloid fibrils. That misfolded architecture can recruit the normal protein and press it into the same conformation. A fold becomes a template. The template becomes a lesson that spreads. Stanley Prusner pushed this protein-only idea in the 198s and 199s and caught heat for it. Without DNA or RNA, how could an agent vary, adapt, or persist? Yet experiments kept stripping away alternatives. Treat samples with nucleic acid, destroying enzymes, and infectivity stayed. Denature the protein and infectivity fell. Step by careful step, purer preparations and transmission studies built a case strong enough for a Nobel. The argument wasn't poetry, it was stubborn residue that refused to die unless protein died with it. If you need handles, take 3. Origami. Once a bad crease exists, it stamps the next sheet. Sourdough. A tiny starter inoculates a batch, and tearing the loaf creates more starter. A corrupted save file. Open it, and the corruption propagates into whatever tries to read and rewrite it. Add the special twist of prions, fragmentation. Long fibrils snap, creating fresh ends where growth accelerates. Growth begets shattering. Shattering begets more growth. Skeptics asked about strains. Without a genome, how do you get distinct types with different incubation times and brain targets? The answer lives in conformers, alternative, stable, misfolded structures of peer-peer. Each conformer exposes side chains and grooves in a unique way, imposing its geometry on new recruits. Pass a mixed population through a host, and selection favors the conformer that converts fastest, traffics better, or resists clearance. It looks like evolution without genes because the selectable information rides in folds. Context pushes too. In vitro, purified PRP can convert with nothing else. In vivo, cofactors tweak the odds. Lipids, polyanions, like RNA fragments, glycosylation patterns on PRP itself, and even metal ions can speed or slow templating. None of that reintroduces a blueprint. It changes the stage, so actors move differently. That helps explain why some brains resist. Chaperones stabilize the healthy state. Certain glycans make docking awkward. Inflammation or aging can tilt the energy landscape so the wrong valley sits a little lower. Zoom into the cell. Misfolded assemblies crowd membranes, disturb trafficking, and drag microglia into a chronic defensive posture. Under the microscope, spongiform change. Tiny vacuoles in the neuropil marks neighborhoods where networks collapsed. Toxicity isn't one note. It's missignaling, membrane injury, synaptic failure, and the slow exhaustion of support cells responding to a problem they can't clear. Meanwhile, labs turn the hypothesis into tools families could feel. Protein misfolding, cyclic amplification, and real-time quaking-induced conversion. PMCA and RT-QuIC offer excess normal PRP as bait, then agitate samples so any seeds recruit and fragment, amplifying a signal a dye can read. In clinics, RT-QuIC on cerebrospinal fluid became a workhorse for sporadic CJD. Some centers validated a simple nasal brushing, reporting around 97% sensitivity for SCJD diagnosis. A fluorescent curve rising on a screen isn't magic. It's the fold caught mid-lesson. Here's the rub on timing. The substrate is PRP itself. Lower it and seeds have less to convert. That's why lowering PRNP expression with antisense tools looks promising in models. It's also why late intervention struggles. Once seeds saturate pathways, stabilizers and blockers push against the tide already breaking into tributaries. Species jumps slot back into the same logic. Sequence differences between, say, bovine and human PRP create friction at the templating interface, the species barrier. Most conformers can't dock well across that mismatch. Rare ones can. Give them enough tries and amplification loops, and a fit appears. Passage in the new host then selects conformers that bind human PRP more efficiently. You didn't mutate a genome, you filtered a shape. One more scene, small but heavy. A junior tech sets up RT-QuIC plates, adds recombinant PRP, pipettes cerebrospinal fluid, seals the wells, and starts the shaker. Hours later, a faint uptick appears on the trace. She runs controls again. The curve holds. Downstairs, a family gets an answer they didn't want. Fast enough to change how a hospital handles steel and how a daughter plans a weekend. Tests don't create seeds. They reveal the lesson already spreading. And that returns us to why shape matters more than temperature or slogans. You can't burn a fold out of existence with routines meant for microbes. You have to respect its routes, contact with high-titer tissues, steel it loves, amplification loops that hand it extra tries, and environments that shelter it. When you see prions as a protein that learns and teaches, prevention stops being mystical. It becomes choreography. Starve the teacher, block the classroom, and don't pass the notebook. Crossing the barrier. Species jumps and strains. While labs proved a fold could teach its neighbors, the world kept asking the harder question, how does a shape in one species find traction in another? The answer sits in a tug-of-war between sequence and structure, the species barrier, and in a cast of prion strains that behave like personalities without genomes. Start with the leap nobody could ignore. BSE in cattle became variant CJD in people. Human and bovine PRP differ at a handful of residues. Those tiny changes make docking awkward, slowing, templating. That friction is the barrier, but it isn't a wall. Feed enough seeds through enough chances, industrial loops, repeated exposures, and a rare conformer finds a way to align. In humans, that docking wrote a young clinical picture. Psychiatric changes first, distinct pathology, and a timeline that traced back to late 198's and early 199's dietary exposure. One fit selected from a noisy crowd stepped through. Here's the plain language model. Think two puzzle sets that almost match. Most pieces don't fit, but if one piece wedges in by chance, it helps the next piece align. That first unlikely fit is the crossing. Passage through the new host then acts like audition rounds. Conformers that bind human PRP better propagate faster. Misfits fade. You didn't mutate a genome, you filtered a shape. Scrapie reminds us the barrier matters. Sheep have carried Scrapie for centuries without clear human transmission in the modern era. Sequence mismatches and the strain repertoire in Scrapie have so far mostly stalled on our barrier. Yet pass Scrapie through rodents with different PRP sequences in the lab, and you can watch incubation times shift and pathology split as selection favors conformers that fit each new substrate. It's evolution's rhythm without DNA. Strains force the identity question. Without genes, what makes one Prion type different from another? Architecture. Misfolded pure P can settle into alternative stable conformations, conformers, that dictate surface features, stability, fragmentation rates, and cell targeting. Those structural fingerprints impose themselves on new recruits, so a strain's look and tempo can persist across passages. In practice, that shows up as incubation differences, brain region preferences, and distinct protease resistance patterns in the lab. A bench story captures the patience it takes to see this. A tech plotted onset curves for mice inoculated with a panel of isolates. First passage looked chaotic, wide ranges, messy lines. Her mentor said, "Don't trust first dates." On second and third passages, the curves converged. The conformer best matched to that mouse's pure P stepped forward. Noise gave way to a clean slope that defined the strain in that context. Tissue distribution writes policy in the real world. Classic BSE and sporadic CJD concentrate highest titers in central nervous system tissues, brain, spinal cord, dorsal root ganglia, eyes, driving specified risk material rules. Variant CJD complicates the map. Lymphoid tissues like tonsils and spleen can carry seeds early, which gave tonsil biopsy diagnostic value and explained why blood transfusion, rare as it was, proved possible in VCJD. The map isn't fixed across strains, it's a travel itinerary shaped by conformation and host biology. At the molecular interface, small details swing big doors. A single residue swap at a key contact can slow conversion. Glycosylation patterns on pure P can shield or expose surfaces. Membrane positioning via the GPI anchor affects how often normal and misfolded forms collide. Some models invoked an auxiliary factor, once nicknamed protein X, to explain cross-species constraints. Others showed that sequence differences plus conformational selection could do the job. In life, both scaffold and neighborhood chemistry matter. Caution belongs here. In vitro amplification tools, PMCA, RT-QuiC, can show that seeds from one species convert human pure P substrate under certain conditions. That's sensitivity, not destiny. A fluorescent curve in a tube lights up possibility. It doesn't guarantee human disease. What it does is tell regulators where not to build amplification loops that give rare fits more tries. Microstory, 20 seconds. In a surveillance lab, a plate lights up when bovine-derived seeds meet human substrate PMCA. The scientist doesn't run to a microphone. She circles 3 words in her notebook. Possible with help. Then she emails the policy team one sentence. Keep SRMs out of food. That's strain talk, meeting public health. Strain behavior also bends clinical storytelling. Variant CJD's pattern in younger patients wasn't random. It reflected a conformer tuned by selection to our PRP and our lymphoid map. Familial mutations shift the substrate beneath the interface, which helps explain why D178N plus methionine at codon 129 steers toward thalamic devastation in fatal familial insomnia, while D178N plus valine drives a different course. Same alphabet, different fold, different itinerary. For families who ask, how did this reach us? The careful answer is, through a rare conformer that overcame a sequence barrier, then refined itself by passage. For farmers and regulators, the takeaway is simpler. Never build systems that hand a rare fit infinite auditions. Close feedbacks, remove high-risk tissues, respect long incubations. Unknown to many city dwellers, out beyond field edges, another Prion has been practicing this math for decades, moving mouth to mouth, hoof to soil, soil to plant, leaving seeds that wait through winter. Its human barrier remains an open file on every wildlife officer's desk. The deer that don't die right. Chronic wasting disease, CWD. Out past the last mailbox, in a stand of aspens, a buck stood broadside and didn't run. His ribs showed. Saliva hung in strings from his jaw. When he finally moved, it was slow and wrong. Legs too long for the ground, he knew. Hunters call it the stare. Wildlife officers call it suspicion. Labs call it chronic wasting disease. CWD isn't new to deer, elk, and moose, but its footprint keeps edging outward on agency maps, county by county, year by year. The stakes land in kitchens that rely on venison each winter, in towns that sell tags and gas and hot coffee to hunters, and in the quiet trust that wild food is clean. Another stake lives in uncertainty. There are still no confirmed human CWD cases. Uncertainty doesn't calm a dinner table. Here's how the shape moves without a slaughterhouse. Infected cervids shed prions in saliva, urine, and feces. Everyday biology turned into a dispersal network. A scrape on a branch, a lick on a mineral block, a puddle in a trail. Seeds slide from mouth to mouth and ground to hoof. Carcasses left on the landscape add a heavier pulse. Scavengers drag tissue. Winter and thaw turn a rib cage into a slow-release packet. Soil isn't neutral. CWD prions bind to minerals, clay especially, and that binding can boost infectivity. Plants can pick up seeds on roots and surfaces, turning a food plot into a reservoir. Months later, a healthy doe noses the same patch and takes a lesson in the wrong fold. A wildlife officer told a story that sets the tone. He followed a GPS collar's death ping to a fence line where a doe lay under Russian olive. When he cut a piece of brainstem and sealed it, the wind carried alfalfa from a nearby field. He burned the tall grass along the fence to clear the area before removing the carcass. One small firewall against a protein that ignores water, winter, and time. Farmed cervids make the map sharper. Close quarters magnify contact. Shared feed and fence line nuzzles multiply chances. One positive test can lock a gate and trigger quarantines. Sometimes whole herds are depopulated. Disposal becomes its own headache. Prions shrug at most landfills and compost piles. High-temperature incineration helps, but access and cost throttle options. Agencies argue over burial depths and liner specs, while communities argue over trucks hauling animals. Nobody wants near their soil. Meanwhile, hunters face a freezer dilemma. A dad in Wisconsin gutted a heavy buck and noticed enlarged nodes in the head. At the registration station, he handed over the nodes and went home to wait. 2 weeks later, the letter came. Positive. He stood in the garage with the chest freezer humming and did the math on grocery bills, then carried the meat to the landfill's designated pit. He didn't need a lecture on absolute risk. He needed a rule he could live with. Science keeps probing the species barrier because policy and panic hinge on it. In vitro assays, PMCA and RT-QuIC, can show CWD seeds converting human PRP substrate under specific conditions. Transgenic mice engineered with human prion protein mostly resist, but some experimental systems hint at cracks depending on design. Macaque studies have been mixed. Methods differ, timelines stretch. In 2024, an NIH team reported that CWD seeds could infect and propagate in human-derived brain organoids under certain lab conditions. That doesn't equal a human case. It does remove the comfort of impossible. So what do agencies say in that gap? They don't soft pedal. Test where programs exist. Avoid high-risk tissues, brain, spinal cord, eyes, tonsils, lymph nodes. Don't saw through the spine. Don't split the skull with the same blade you'll use for steaks. Wear gloves. Keep designated knives for field dressing. And deboning. If a test comes back positive, don't eat the meat and don't feed it to pets. These aren't theater. They add friction to a pathway prions prefer. Hunters adapted faster than some experts expected. Check stations that once felt like a hassle now run like pop-up rituals. Coolers open, tags scanned, nodes snipped, advice traded. A teenager in blaze orange watched a biologist press forceps into a jaw hinge and asked, "Is it safe?" The answer was exact. "We're managing risk until we know more. Get your deer tested. Avoid the head and spine. Wait for results." The kid nodded like he'd been handed a secret. It wasn't a secret, it was a system. Policy lags and leads at once. States roll out carcass transport rules. Don't haul whole heads across county lines. Leave spinal columns where you harvest. Bans on baiting and feeding reduce nose-to-nose contact. Farm cervid surveillance tightens. Movement permits stack paperwork on fence posts. Not every change is popular. Some feel like punishment for problems born miles away. Disease doesn't care about blame. It cares about contact and time. If you want a pocket map, carry this. Test where programs exist. Don't consume meat from a positive animal. Keep brain and spinal cord out of your grinder. Bag and dispose of remains through approved channels. Ask processors how they segregate and clean. Share results with neighbors. None of it guarantees an outcome. All of it lowers the number of lessons a bad fold gets to give. Back in the aspens, the buck with the stare finally stepped and melted into shadow. The hunter lowered the rifle and checked tag dates on his phone. Somewhere between the trees and the lab, between policy and a plate, we're writing the answer to a question we'd rather not ask. How many chances will we give a prion to try us? The needle and the vein. Blood, transfusions, and screening. Hospitals sleep with one eye open because blood doesn't wait. When a trauma rolls in or chemo drops counts, someone needs red cells now. That urgency met Prions head-on in the variant CJD years, and for a few patients, the answer to, can it spread in blood, was yes. The story surfaced quietly. A man in the United Kingdom got a routine transfusion in the late 199s. Years later, he developed variant CJD. Tracing records showed his donor later died of the same disease. Another recipient, different ward, same thread. The route didn't explode into an epidemic, but it punctured a hope. Prions in VCJD can ride blood, even at levels too low for ordinary tests to catch. Universal leukodepletion, stripping white cells, was added broadly for general safety. It may reduce risk, but it's not a force field. Policy had to move. Across a city, a would-be donor squeezed a stress ball and watched the bag fill, until the nurse checked his travel history. He lived in Britain during the BSE era. Deferral. He left with a cookie he didn't want, feeling pushed out of a civic ritual. The rule wasn't personal. It was a hedge around uncertainty. Geography and time-based deferrals became blunt tools that protected recipients and quietly strained supply. Meanwhile, in a hematology clinic, a man with hemophilia asked a question that flattens rooms. Is my factor safe? Plasma-derived products had already weathered other infectious crises. For prions, regulators and manufacturers tightened, sourcing, refined fractionation, and stacked safety steps. No blood-based prion screening test made it to mass deployment without trade-offs that hurt more than they helped. False positives aren't abstract. They pull safe units from shelves, scare donors, and ripple into operating rooms. What makes screening brutal? Prionemia, the seed level in blood, can be vanishingly low, scattered across plasma, cells, and tiny vesicles, and pattern strains can shift. You're hunting whispers. Lab tools born from prion biology amplify whispers. Protein misfolding, cyclic amplification, and real-time quaking-induced conversion. PMCA and RT-QuIC. Flood samples with excess normal PRP. Then agitate so any seeds recruit and fragment, lighting up a dye that loves beta sheets. In clinics, RT-QuIC found its lane diagnosing sporadic CJD from cerebrospinal fluid, and in some centers from a simple nasal brushing with reports around 97% sensitivity. But transfusion safety is a different mountain. You can't lean on spinal taps. You need high throughput, near perfect specificity, and sensitivity strong enough to catch a seed without torching the blood supply with false alarms. Policy whiplash followed the science in fits. Deferrals widened, then narrowed as data matured. Some countries later eased blanket bans on donors with remote UK residency. Others held the line. Surveillance expanded in the background. Tonsil and appendix archives were sampled for silent VCJD markers to estimate background exposure. Numbers stayed low, not zero. Each tweak balanced the visible harm of short blood supply against the invisible harm of a missed prion. Here's a micro story that shows the edge. A woman scheduled for cardiac surgery got a call the night before. The hospital had enough O-negative for emergencies, but elective cases were delayed to conserve stock after an upstream recall. The recall had nothing to do with prions, but it proved how fragile supply is. Policies that defer thousands of donors to trim a rare prion risk echo into rooms where timing is life. Ethics sits at the center. Imagine a prion screen that's 99.5% specific. Across millions of donations, that 0.5% means tens of thousands of safe units destroyed and donors labeled with a stigma no one can clarify. Meanwhile, true positives might be so rare you never confirm one in real time. Regulators ask the hard question: does this net harm exceed the risk we're managing? Confirmed transfusion transmissions happen with variant CJD. For sporadic CJD, transfusion transmission hasn't been confirmed. Measures instead focus on instrument handling and tissue risk, not mass blood screening. So where do we land? Donor deferrals by geography and time for VCJD risk. Universal leukodepletion in many systems. Vigilant hemovigilance, linking donors and recipients when a case appears and tracing lookbacks. Research that keeps pushing amplification assays closer to field use, while clinics use RT-QuIC where it shines. Faster diagnosis for patients already ill, guiding care, and triggering Prion protocols for instruments before exposure multiplies. There's a note taped above a bench at a blood center that sums up the posture. Speed matters, so does certainty. That's the needle in the vein. Move fast enough to save the patient in bed 5, and slow enough to keep a fire from hitching a ride to bed 6. Sterilization limits. Inside the hospital, cleaning was supposed to be the boring part. Heat, pressure, chemicals, repeat. Prions turned that routine into a trap. They don't carry genomes you can shred with UV. They don't fall apart when you flood them with the usual enzymatic detergents. They hold their fold like a grudge, and they love stainless steel. Stakes first, because this isn't theoretical. One slip can turn an operating room into a months-long question mark. Budgets bend when entire trays get tossed. Surgeons delay cases. Patients receive letters that tilt dinner tables. Certainty costs money. Uncertainty costs sleep. Action looks like this. A biopsy comes back. Probable prion disease. Infection control rewinds the tape. Which room, which tray, which devices touch brain, spinal cord, or the posterior eye? Standard reprocessing. Pre-clean, washer disinfector, autoclave. Doesn't guarantee prion inactivation. Seeds cling to microscopic scratches in steel, and the very heat and steam that rescue us from bacteria don't reliably erase a folded template. Some disinfectants even cross-link proteins, gluing seeds to surfaces. The fix is ugly but effective. Heavy sodium hypochlorite, or 1 to 2N. Sodium hydroxide soaks for defined times, followed by prolonged autoclaving at higher temperatures. And even then, many teams choose destruction over reuse. Microstory, 20 seconds. A sterile processing lead gets a 10 p.m. call. Suspected prion in yesterday's neuro case. She pulls the tray log, isolates a shelf, and drops 3 micro-instruments into a red Prion bin marked for disposal. Thousands of dollars vanish in a clatter. She signs the loss and sleeps that night. Timing rules everything. Dried organic residue shields seeds from contact with chemicals. That's why the clock starts the second an instrument leaves tissue. Keep it wet. Get it down. Flush lumens now. Those mantras sound fussy until you realize a smear of dried protein can protect a teacher seed from your best efforts to evict it. Guidelines evolve the hard way. If prion risk is on the table, certain neurosurgeries, posterior eye procedures, work that may contact lymphoid tissue in suspected variant CJD, hospitals isolate instruments, prefer single-use where possible, and, if reprocessing is unavoidable, follow prion-specific protocols in segregated workflows. Color-coded tags, prion-labeled bins, and laminated algorithms sit at eye level so nobody drops a suspect rongeur into a standard washer that would then contaminate the chamber. Documentation is religion. Device exposure, cycle parameters, lot numbers, so decisions take hours, not days. Meanwhile, steel isn't neutral. Microscopy shows seeds adsorb to stainless in ways rinse cycles don't undo. Some research points to copper surfaces being less welcoming, but you don't swap a hospital's inventory on a hunch. What changed instead were behaviors. Simplify instrument sets for high-risk cases, use single-use suction tips and burrs that touch neural tissue, and quarantine complexity you can't trust. Vendors now flag prion compatibility for materials. That small icon on a spec sheet prevents big arguments after midnight. Ophthalmology learned a separate lesson. Variant CJD can involve lymphoid-rich tissues early, and ocular tissues may carry risk. So posterior eye work in a patient with concerning history triggers single-use where possible and prion-grade handling for anything that crosses the field. GI units wrote contingencies too for lymphoid-heavy procedures if variant exposure is suspected. These carve-outs slow throughput and burn money. They buy certainty. Here's another micro-story. Tuesday, 2 a.m. A posterior eye case with odd travel history hits pre-op. The charge nurse tags the cart Prion Precaution in red. The scrub tech swaps in a sealed single-use kit. The reprocessor tapes off a bay. An extended 134 degrees Celsius autoclave run gets scheduled for the few reusables that cross the drape. In the morning, administration asks about cost. The supervisor slides an incident sheet across the table. Cheaper than a letter to 200 patients. Impact ripples beyond steel. ORs pause rooms for decontamination. Schedules reshuffle. Finance signs off on losses measured in trays, not tools. Most notified patients will never get sick. True exposures often evaporate under review. But anxiety doesn't wait for nuance. Hotlines open. Nurses repeat careful sentences. What might have happened, what we did, what to tell providers in the future. Not every chapter ends bleak. The switch from cadaver-derived materials to synthetic grafts closed an old road. Recombinant hormones erased an entire iatrogenic stream. Education moved upstream. Residents now learn Prion pathways the way they learn hand hygiene. Labs fast-track RT-QuiC when a rapid answer could prevent an instrument chain from becoming a liability, even knowing diagnosis will outpace any treatment. The meaning lands quietly. Fire alone doesn't clean this problem. With prions, cleanliness is choreography, pathways, timing, segregation, and the discipline to throw away what you can't trust. A tech at end. Whispers before the storm. Early detection and biomarkers. If fire can't fix steel, then time becomes the only lever we still control. Early matters, not because we can stop the disease yet, but because we can steer care, protect instruments, and spare families weeks of spinning in the dark. For years, we read damage instead of the seed. Cerebrospinal fluid markers like 14, 3, 3, and tau climb when neurons died. Useful, but blunt. They said, something's breaking, not a template is teaching. Clinicians wanted a microphone pressed to the fold itself. In a dim room with a shaking plate, that microphone arrived. Real-time quaking. Induced conversion. RT-QuIC doesn't ask about cell death. It asks whether a seed can recruit. Technicians add a surplus of normal recombinant prion protein to a patient sample, usually spinal fluid, sometimes a nasal brushing, then cycle shake and rest. If seeds are present, they grab substrate, convert it, fragment into more ends, and grow again. A dye that loves beta sheets lights up as fibrils form. On the monitor, a flat line arcs upward. That curve isn't rumor, it's templating, caught mid-lesson. Speed meets families where they live. Instead of waiting weeks for a maybe, Neurology can deliver clarity fast for sporadic CJD. Some centers now use a simple nasal brushing to sample the olfactory epithelium and report around 97% sensitivity for SCJD. A nurse in a quiet room explains it gently. We're detecting seeding activity. It isn't magic, it's amplification of what was already happening in the brain. Micro story, 20 seconds. A lab team watches a faint fluorescence trace finally cross threshold. The tech checks controls twice, then lowers her head. Downstairs, a social worker walks a spouse through power of attorney forms and flights for adult children. Results don't rescue. They give honest time. Meanwhile, amplification tools widen surveillance. RT-QuIC and its cousin, protein misfolding cyclic amplification, PMCA. Let researchers ask softer questions with loud consequences. Are variant seeds present in lymphoid tissues like tonsils earlier than we thought? Do archived appendixes whisper about silent exposure in a population? Each positive in a repository isn't a diagnosis for that person. It's a data point that shapes blood donor deferrals, instrument protocols, and budget lines for monitoring. It's how public health listens for a train before it rounds the bend. Specificity matters as much as speed. A false positive in diagnosis crushes a family. A false positive in screening can drain a blood bank and shutter operating rooms. That's why labs live by negative controls, replicate runs, kinetic fingerprints, and confirmation across independent plates. Borderline curves don't become phone calls without context. The goal isn't fast alone. It's fast enough and right enough to act. Early results can reroute steel. Picture a patient scheduled for a diagnostic brain biopsy, a procedure that would saturate neurosurgical instruments with the highest risk tissue. An urgent RT-QuIC, positive the night before, flips the plan. The team cancels the biopsy that would have sacrificed expensive trays, or if they must proceed, activates Prion-grade protocols from the first incision. One fluorescent curve upstream spares steel downstream and prevents 100 anxious letters. Nasal brushing surprised people because it felt too easy. A small flocked swab gathers cells from the olfactory epithelium. No lumbar puncture, no sedation, and yet reveals seeding in the central nervous system in SCJD at reported high sensitivity in experienced hands. Technique and validation matter. But for some families, The road to an answer runs through a clinic chair instead of an operating room, and that swap is mercy. Seed amplification also powers drug development, even in a field short on drugs. Candidates that aim to stabilize normal PRP, block interfaces, or deplete substrate can be tested against patient-derived seeds in vitro. If a compound flattens an RT-QuIC curve or slows PMCA kinetics, it earns another experiment. Cells, then animals, then maybe a phase 1. The same microphone that diagnoses becomes a metronome for therapy work. Biomarkers don't stop with a single assay. MRI diffusion-weighted imaging draws bright signatures in cortex and basal ganglia for SCJD. EEG may show periodic sharp wave complexes in the right window. Blood-based markers of neuronal injury like neurofilament light sketch disease intensity. None replace the seeding assays. Together they build a mosaic that trims uncertainty and accelerates decisions. Here's where ethics breathes. Families ask, does testing make it worse? The answer is clean. No. RT-QuIC doesn't create seeds. It copies their effect in a tube to reveal what's already there. That distinction keeps labs from becoming scapegoats and lets hospitals use results to activate Prion protocols without fearing they nudge the domino. Public health stitches these whispers into real changes. Faster confirmation sharpens surveillance curves, flags iatrogenic risk sooner, and justifies Prion-grade sterilization when it counts. In chronic wasting disease country, amplification assays inform clear messaging. Test in affected units, avoid high-risk tissues, wait for results. And always the honest line, there are still no confirmed human CWD cases, even as 2024 organoid data and in vitro work demand vigilance. Final human moment. A clinician draws two slopes on paper, one steep and short, one flatter and longer. We can't change this curve yet, he says, tapping the steep line, but because we know sooner, we can change what happens under it. He lists choices: hospice early, speech therapy now, a last road trip this weekend, or protocols that won't turn one tragedy into many. Early detection isn't a cure. It's a way to spend the time you still own, and a way to keep steel and blood from carrying a lesson that never should have had a second student. Fighting a shape, therapies that almost worked. Detection sprinted ahead, treatment limped. Families brought printouts to clinics and asked for anything that might slow a fold already teaching. What they got were almosts. Good ideas that moved curves and dishes, then crashed into timing, delivery, and the geometry of a brain already under siege. Quinacrine looked like a doorstop you could swallow. It binds proteins, and in cell assays, gummed up the interface where misfolded PRP recruits the healthy form. Compassionate use stories spread. A few patients seemed to level off for weeks. Hope outran data. Larger, controlled attempts didn't show durable benefit. The molecule hit targets in dishes. The human blood-brain barrier, distribution, and toxicity windows told a harder story. Pentosan polysulfate tried a different lane. Flood the cerebrospinal fluid directly. Surgeons tunneled intraventricular catheters and infused the sugar-like polymer to block conversion. One or two patients outlived grim expectations. Others didn't. Complications stacked up. Bleeding, infections, pressure issues, and benefit wasn't consistent. Bathing every synapse in a uniform dose proved more fantasy than protocol. Doxycycline stepped in from the antibiotic aisle, buoyed by reports it could destabilize fibrils or blunt seeding. An early Italian study teased survival signals when given to people at risk or very early. Broader trials across sporadic cases didn't repeat the win. Timing kept stealing the ball. By first symptoms, seeds had already threaded along networks. Untangling that lattice with a late-arriving small molecule was like trying to unbake a loaf. Antibodies felt inevitable. Target PRP's surface, block templating, flag aggregates for cleanup. In engineered animals, some antibodies slowed disease if given early enough. Translating to people hit two walls. First, distribution, getting enough antibody across the blood-brain barrier without provoking inflammation that worsens injury. Second, target safety. The healthy form of PRP lives everywhere in the brain. Cross-linking it at the wrong epitope can be toxic. Doses high enough to matter flirted with danger. Doses low enough to be safe didn't move trajectories. Then came the cleanest logic in years. Starve the template. Seeds need substrate, PRP. Lower it, and conversion slows. Antisense oligonucleotides, short strands that bind PRNP mRNA and dial down PRP production, delayed onset in models when delivered before or very early after seeding. The catch was the same brutal clock: intrathecal delivery, repeated dosing, and a disease that declares itself when highways of spread are already busy. Designing trials for a sprinting neurodegeneration is like building a runway after the plane's airborne. Small molecules circled back with new tricks. Some aimed to stabilize the normal fold, nudging the energy landscape so the alpha helical state stayed favored. Others tried to cap fibril ends, business tips where conversion happens, turning seeds into dead twigs. A few targeted cofactors, tweaking lipids or polyanions that grease templating. Hits in RT-QuIC or PMCA, flatter fluorescence curves, slower kinetics, earned animal studies. Many signals shrank with each step closer to people. Here's the corridor story from a halted study. A coordinator sat with a family whose loved one just missed the dosing window when the safety board paused the trial. No clear benefit, worries about adverse events. The mother asked, "Can we keep going anyway?" The doctor said no, then pivoted to what still mattered: sleep, swallowing, pain, a weekend with grandkids, while words still held. The word help felt small. It was also real. Delivery haunted every bright idea. Convection-enhanced delivery could push drugs through brain tissue, but only locally and with risk. Intrathecal routes spread agents through cerebrospinal fluid, but parenchymal penetration was patchy, and dosing collided with frailty. Even when a compound reached its target, the disease's fastest downhill often began just as enrollment paperwork cleared. Strategy started sharing oxygen with chemistry: lower the substrate, block the interface, clear assemblies, layered, not single shot. Use seed amplification as a pharmacodynamic gauge. If a candidate mutes an RT-QuIC curve fed with patient cerebrospinal fluid or slows PMCA growth, it earns a longer leash. Pair that with biomarkers, diffusion MRI, neurofilament light, to see if the biology budges in life, not just in a tube. Trials began creeping earlier, looking to catch the whisper phase, not the siren. The field also learned to tell the truth in ways families could carry. A father said, "Give me 3 good weeks, not 3 bad months." And clinicians wrote that into goals. Start speech therapy now, not next month. Treat insomnia before nights shatter. Manage pain before panic. Hospice early as a structure for control, not surrender. The absence of a cure didn't mean the absence of care that changes days. Almost weren't wasted. Each one mapped a boundary: how much substrate you must lower, how fast you must arrive, which epitopes you can't touch, what the blood-brain barrier will and won't forgive. The likely future won't be a miracle headline. It'll be a layered defense started early. PRP lowering to starve seeds, small molecules to stabilize or cap, immune tools to clear debris, guided by assays that listen to the fold in real time. Hold one last lab-to-bedside thread. The same fluorescent curve that diagnoses a family's worst day can also mark progress in a vial as a candidate drug flattens its rise. It's not victory yet, it is a ruler. And rulers, used honestly, are how sprints become marathons. And how marathons one day slow to walk someone can live with. The Prion shadow: Alzheimer's, Parkinson's, and beyond. Treatment stalled on prions, but their rulebook leaked into clinics that thought they were fighting different wars. Same physics, slower clock. A misfolded shape teaching neighbors, not in months, but across decades. Start with the rooms where memory thins by inches. In Alzheimer's, beta amyloid piles into plaques and tau twists into tangles. For years, we treated them like trash left by dying neurons. Then labs seeded healthy brain slices with a dusting of misfolded A-beta or tau, and watched the same chain reaction we met with PRP. Seeds recruited normal protein, built fibrils, fragmented, and spread. In animals, inject a tiny tau seed in one region, and months later, pathology crept along the brain's wiring, hippocampus to cortex, station to station. The network wasn't scenery, it was a railroad. Parkinson's traced its own shadow with alpha-synuclein. Small seeds in culture, or rodent brains, coax normal alpha-synuclein into Lewy bodies and Lewy neurites. Pathology then marched from gut and olfactory areas toward deeper nuclei. Patients who'd lost smell years before tremor weren't curiosities. They were a map to the first embers. The gut-brain axis stopped being a slogan and turned into mile markers. Enteric nerves holding seeds, vagus pathways offering routes upstream. Epidemiology that linked truncal vagotomy with altered Parkinson's risk didn't make a therapy, but it drew an arrow. Clinicians started saying a sentence that sounded like Prion science with new nouns. It spreads. Not person to person, but as a conformation along connected tissues. That shift forced Prion-style questions. Are there strains, distinct amyloid, tau, or alpha-synuclein conformers that steer disease tempo and terrain? Structural biology answered yes. Cryo-M showed polymorphs, different knots tied with the same rope. Some convert faster, some target different cells, some resist clearance. Suddenly, patient-to-patient variability had a physical face. Here's a micro-story that lands the pattern. An autopsy tech who'd handled CJD cases for a decade watched a series of Alzheimer's brains and muttered, "They're following the roads." The pathologist didn't laugh. He rolled out connectivity maps and traced the same lines: hubs first, long-range tracks next, capillaries of spread last. The shape taught, the network listened. Medicine's mirror added a hard glint. A handful of case series described adults who developed cerebral amyloid angiopathy, amyloid choking brain vessels, decades after receiving cadaver-derived pituitary products or certain childhood neurosurgical grafts. These weren't prion diseases. They were ABA pathologies in relatively young adults. Numbers were tiny. Debates about causation were loud. The lesson was a whisper, not a verdict. Seeds beyond pure P can hitch rides on steel and biologics. Surgical societies began asking if tools that touch amyloid-rich tissues deserve extra caution. Not because routine spread is likely, but because prion history already taught humility about unlikely. Labs repurposed prion microphones. RT-QuiC variants tuned to alpha-synuclein began catching seeds in cerebrospinal fluid and skin biopsies from people with synucleinopathies, sharpening early diagnosis. Seed amplification for tau helped separate Alzheimer's from frontotemporal lobar degenerations when MRIs blurred. The assays didn't invent disease. They put ears to tracks that rumble for years before daily life notices. Therapies took notes and hits. Antibodies against amyloid rarely reversed established plaques, but some began to slow decline modestly when given earlier. Side effects and costs sparked fair fights. Supporters pointed to curves that bent a little. Critics pointed to small wins with heavy price tags. The shadow of prion logic cut through the noise. Timing is ruthless. Once networks reroute around damage, clearing aggregates won't rebuild roads. Intervene when seeds are whispers, not shouts. Parkinson's trials nudged in the same direction. Alpha-synuclein antibodies aim to mop up extracellular seeds and blunt spread. Results have been mixed. Delivery, dosing, and target engagement fought each other. Meanwhile, clinicians leaned harder on strategies that respect spread. Exercise programs that stabilize circuits, sleep, and gut care to quiet inflammations that tilt folding, early speech and swallow therapy before traffic jams become sinkholes. No silver bullet. Many small detours. The gut-brain thread invited everyday agency. Diets that reduce chronic inflammation, constipation management that keeps enteric nerves calmer, microbiome research that, while early, suggests some communities may dial the fuse. None of this is a cure. Each step is friction against the classroom where misfolds teach. Hospitals adjusted posture too. No one is equating Alzheimer's to CJD, but instrument handling got sharper in narrow windows. Amyloid-heavy neurosurgical work or pathology labs that process plaque-rich tissue. Because pathways matter as much as pathogens. The prion lesson isn't panic, it's respect routes. Here's a human scene from a memory clinic that stitches science to choice. A neurologist lays out two tools, a tau PET scan and a seed amplification assay tailored to tau. The patient asks, "Will this fix me?" The doctor answers, "It won't. It tells us which road you're on and how fast you're moving, so we can pick turns you'll feel." That means planning speech therapy now, not later. It means stabilizing sleep to protect fragile circuits. It means trials that fit the biology on their terms, instead of hoping a late antibody can unspool years of teaching. What Prions gave these fields wasn't a scare tactic. It was a language. Strains without genes, seeding without life, spread without blood. When you see disease through that lens, prevention becomes choreography again. Starve the substrate, block the interface, cool the terrain, and medicine becomes quieter and braver at the same time. Less about chasing monsters, more about closing classrooms with the wrong shape.
These tricks and evolution's odd bet.
After an hour of shapes that ruin minds, here's the twist. Some cells use prion-like switches on purpose. Not as accidents, as memory. In a warm brewery, one stubborn yeast line kept a richer head and shrugged at salt. The brewer swore it had personality. Down the road, a grad student streaked a single Saccharomyces cerevisiae strain and watched colonies bloom in different colors from identical parents. No DNA change explained it. She was staring at inheritance written in folds. Yeast carry prion-like elements. Paralinguistic PSI+, paralinguistic URE3 paralinguistic, and others that flip traits by changing how proteins assemble. Take paralinguistic PSI+, paralinguistic. A protein called SUP35 helps ribosomes stop at the right place on mRNA, periods in the genetic script. When SUP35 self-assembles into amyloid, much of it gets sequestered. With less SUP35 free, stop signals occasionally get ignored, and ribosomes read through, adding tails to proteins. Most days messy. On some days, high salt, new carbon sources, those extended proteins help. The conformation becomes a survival shortcut. The cell can pass to daughters by handing them a sliver of the SUP35 aggregate at division. No mutation required. It's epigenetic memory written in shape. Tells a parallel story. URE2 normally represses genes that scrape nitrogen from lousy meals. Flip URE2 into amyloid, repression eases, and the famine program turns on. Costly in feast, life-saving in famine. Because the amyloid templates itself, the on state inherits, until the assembly dissolves and the trait vanishes. Fast to switch, fast to forget. Here's a micro story that nails the mechanism. The grad student plated her yeast with and without guanidinium chloride, which inhibits the chaperone HSP104. On plates with guanidinium, the colony colors stabilized. On plates without it, they flipped. HSP104's job is to shear amyloid fibers into transmissible seeds. Block the scissors, and the prion state can't spread well. She wrote one sentence in her notebook: memory needs scissors. Why would evolution risk this? Because mutation is slow and permanent. Conformational switching is fast and reversible. In fluctuating environments, a population hedges its bets. Most cells keep the safe fold. A minority flips, paying a cost today for a shot at tomorrow. If conditions change, that minority seeds the next day's majority. Diversity by conformation lets a clonal population act like a portfolio. This isn't lab-only weirdness. Wild yeast isolates carry these elements. Some prion-like states tune metabolism. Others influence adhesion and biofilm-like behaviors. Microbial communities glued to surfaces by functional amyloids. That word amyloid sounds like doom in neurology. In cell biology, structure decides friend or foe. Our own bodies use ordered protein assemblies for work. Pigment organelles, scaffolded by functional amyloid-like fibers. Peptide hormones stored densely in secretory granules, signaling hubs that phase-separate into gels during stress. Shape carries information. The trick is control. Assemble on cue, disassemble on cue. So does this mean our brains run on good prions? Careful. Neurons rely on transient assemblies, synaptic proteins that condense and disperse, stress granules that form and clear. There are hints that some long-term cellular states lean on self-templating assemblies, but the line between functional assembly and runaway amyloid is thin. Long-lived cells like cortical neurons can't afford a switch that sticks. What's an adaptive bet in yeast can be a slow catastrophe in us. Evolution didn't invent evil for humans and elegance for microbes. It reused physics. The same rules that let a deer prion cling to clay and persist also let a yeast cell remember famine without a mutation. Context sets the price. Short-lived, rapidly dividing cells can play with folds and cull mistakes. Long-lived, complex circuits pay dearly when a fold won't let go. Hold a small scene from the brewery. The brewer skims foam, tastes a batch, and says, "Practical shields, personal and public risk. Bring this home." The point isn't dread, It's friction. Small, deliberate moves that make the easy path for a prion the path it never takes. If you hunt in chronic wasting disease country, start with a checklist. Check your state's map before the season. If your unit offers testing, pull retropharyngeal lymph nodes at the registration station or have a biologist do it. Then wait for results. Field dress with gloves. Keep separate knives for skinning and deboning. Don't saw through the spine. Don't split the skull with the same blade you'll use on steaks. Bag the head and spinal column for approved disposal, incineration, or designated pits, not the backlot. If results come back positive, don't eat the meat, don't feed it to pets, and don't run it through a grinder that will touch other animals. One careful week in November protects dozens of dinners. At the processor, ask 3 plain questions. Do you segregate carcasses? Do you clean grinders between animals? Can you process mine alone? If they shrug, find another shop. If they show you logs and a cleaning routine, stick with them and tip well. At home, label cuts, keep brain and spinal cord out of the grinder, and clean tools with hot soapy water first. Detergent removes organic residue that can shield seeds, then follow your local guidance for disposal. Don't compost heads in CWD areas, grocery aisle rules are simpler. Bovine spongiform encephalopathy safeguards still stand. Ruminant protein isn't fed back to ruminants, and specified risk materials, brain, spinal cord, dorsal root ganglia, are kept out of the food chain. You benefit from those policies without thinking about them. If you buy novelty products that include head or spine, know the source or skip it. Hospital side, you get to ask 1 sentence before high-risk procedures. Do you have prion protocols? You're not accusing anyone. You're checking that there's an alert pathway, if rapidly progressive dementia is on the table, or if surgery might touch high-titer tissue. For neurosurgery and posterior eye work, single use where possible is worth the price tag. If you ever receive a letter about a potential exposure from old dura mater grafts or past instrument concerns, scan it, keep a copy with your medical record, and bring it to future procedures so teams can choose instruments and sterilization steps that don't turn one mistake into many. In labs, post prion SOPs where hands live, not hidden on a server. That means prion-labeled waste bins, secondary containment for sodium hypochlorite or 1 to 2N sodium hydroxide soaks, defined exposure times, and documentation before disposal. Wear face shields and cut-resistant liners under nitrile. Keep tissues wet. Dried residue shield seeds. If a suspect sample touches shared equipment, quarantine first, argue later. RT-QuIC can move fast, but it's not a reason to relax containment. It's a reason to activate it sooner. Families need clarity more than drama. If a loved one is diagnosed or strongly suspected, tell every provider who might use instruments, dentist, ophthalmologist, surgeon, so they can adjust. Hospice early is control, not surrender. Pain, sleep, speech, swallowing, and quiet legal tasks handled before they're urgent. If a familial mutation like D178N is in play, ask for genetic counseling and decide, as a family, what to share and when. Public steps multiply private ones. Support state funding for CWD testing and carcass disposal. Those pits and incinerators are the difference between one freezer and a whole town's risk. Back blood policies that balance supply with safety, geography, and time-based deferrals feel personal, but they protect strangers on operating tables. Vote for hospital budgets that include sterile processing staff and prion-compatible workflows. Quiet line items keep hotlines quiet. Micro story, 15 seconds. A teenager in blaze orange watches a biologist clip lymph nodes and asks, "Is it safe?" The answer is exact. We're managing risk. Test. Avoid brain and spine. Wait for results. He nods, then shows his dad how to bag the head. One last rule of thumb you can carry without a lab coat. Pathways, not panic. Don't create amplification loops. Don't move high-risk tissues into places they don't belong. Don't give a rare fit unlimited auditions. Friction wins when it's boring, repeated, and shared. Return to the bedside. Closure and meaning. She'd been the nurse who could fix an IV pump by sound and calm a panicked family with a hand on a shoulder. The day she laughed and couldn't stop, her coworkers told each other it was stress. A week later, she stood in a corridor. She'd walked for years and asked which way led to radiology. Another week, her swallow began to fail. The team she trained now spoke softly outside her door. Diagnosis didn't arrive with a single trumpet. It came as a stack of careful sentences. The MRI showed diffusion signals in places that write habits into days. The EEG traced disordered rhythms. In a dim room down the hall, an RT-QuIC curve began to climb, reporting seeding in clear fluorescence. A neurologist sat with her family and said the word they tried to dodge. They didn't cry then. They wrote down instructions. They chose who would sign papers when she couldn't. At home, rituals changed shape. Her daughter labeled cabinets, plates, bowls, tea. A neighbor left soup on the porch. Her husband moved a chair to the bedroom window so she could watch the maple turn from green to rust to bare. On a good morning, a friend from the ward sat at the bed's edge and read chart notes the way they used to trade night shift stories. They laughed once, a small sound that didn't spill into something else. Her hospital took the case personally. Infection control retraced every instrument that had touched her care before the diagnosis, then tightened the Prion protocol everyone thought they already knew. A red Prion, been moved from a back shelf to the front of the room. The sterile processing team posted a laminated flowchart where no one could miss it. Neurosurgery updated a checklist so residents at 2 a.m. would know which sets to quarantine, which to discard, and which to run through the long decontamination they'd memorized but never used. It wasn't flashy. It was a dozen small decisions that meant the next family wouldn't get a letter months later. The blood bank added a standing note for rapidly progressive neurologic cases. Hold implicated units until confirmation or clearance. It made scheduling harder. It also answered a question staff had been whispering since the first transfusion alerts in variant CJD years. What does caution look like when stakes are invisible? Her last months didn't become heroic. They became precise. Speech therapy arrived early, before words scattered, teaching her husband single-syllable prompts and quiet pauses that create space. The palliative team tuned sleep and pain before either unraveled, then moved faster when nights shortened. Friends visited 2 at a time, no more, and sang the simple songs she loved from high school choir. One afternoon, they all stood in the kitchen with mugs that went cold and admitted on purpose what they'd been avoiding. It didn't change a lab value. It did give them a day without pretending. In the end, the unit she'd mentored pulled a night shift in her living room so her family could sleep. She died before sunrise, the maple outside black against a silver sky. Later that week, the OR ordered single-use kits for posterior eye cases and updated an email that goes to charge nurses when a Prion protocol activates. No one outside noticed. Inside, people who understood watched the large system move an inch the right way. What stayed with them wasn't a twist solved. It was the shape of the thing they'd been chasing for 2 hours. The way a fold can unwrite a life, and the way a dozen unglamorous choices can keep that fold from traveling farther than it has to. Her husband left the chair by the window for a while. In spring, the maple budded green. At the hospital, the red bin stayed where it was, easy to reach. That bin wasn't fear. It was respect, for pathways, for timing, for the fragile geometry we live inside every day. A protein with no genes taught us a hard rule. Shape can unwrite memory, so respecting routes matters as much as finding cures. If you want more hidden links between prions, hospitals, food, wildlife, and the tech we trust, subscribe now. Then watch our follow-up on medical mysteries we can actually fix. And share this with a hunter, a nurse, and someone who still thinks sterile means simple.